In this video we will discuss how this light scattering property is related to the melanin concentration. So this is the image of the cells that are differentiated. So at the bottom is the melanoblast cells derived from H9SESC cell lines, and top one image is the differentiated cells, that is melanoblast differentiated into melanocytes, and the figure on the left sides are on the phase diagram and figure on the right sides are the TEM diagram. Here we can see the melanocytes can be seen in darker in color both in the phase diagram and also in the TEM diagram. So, and later on did the when they analyze the light scattering property through side scatter of the property of the flow cytometers, they can see the difference in the side scatter during the initial days of the differentiation and during the uh, end of the or in the final stage of the experiment, that is during the 25 days. They can see the melanoblasts are more, so differentiation is not happened that much. After that, when they reach around 45-62 days, they can see the differentiation has increased as and it can be seen with the increase in the side scatter signals in the melanocytes, and these results are also in line with the phase images and TEM images of the differentiated cells. So from this they made a hypothesis that the side scattering properties of the cells could indicate the melanin concentration. Thereafter, they also plotted a linear relationship between side scatter and the melanin concentration, for which they got R square value is around 0.823 and P value is around 1.8810 to power minus 6. And after that, what they did is they check whether these side scattering properties are really they check their hypothesis whether the side scattering properties are really because of the melanin concentration or something else. So in order to test that hypothesis, they knocked out the gene that is responsible for upregulation of melanin, or we can say that is the melanogenic gene, that is tyrosine, that is TYR. And in the control experiment they can see the melanin concentrations, the melanin side scatter properties are higher, and when they knocked out the gene, and they have used 4 different guide RNAs. So when they knocked out those when they use those guide RNAs to CRISPR knockdown this tyrosine, they can see the reduce in the side scattering properties, and when they re-express those gene, they can see some increase in the side scattering properties. So from this they further substantiate their hypothesis that melanin properties is related to the side scattering. Melanin properties are the one of the indication for the side scattering properties are one of the indication for melanin concentration. And the later on they quantify this side scattering signals using box plot, and they can see that when in the control experiment they can see the high level of melanin, whereas when the tyrosine is knocked down they can see the reduced level of side scatter. They can see the reduced level of side scatter, not the melanin. The y-axis represents the side scatter. So in the control they can see the high signals for side scatter, and when the tyrosine is knocked out they can see the low expression of low signals for the side scatter, and when the tyrosine is re-expressed they can again see the high signals for high or increased signals for the side scatter. And the color within the dots represents the concentration of melanin. So here the concentration of melanin in the control is high, and in the case of tyrosine the melanin concentration is low, and in the case of this re-expression of tyrosine melanin concentration increases, and the scale is represented here. So later on, when they quantify this melanin in this microgram per ml in the different cell system, they can see the melanin concentrations are in line with the side scatter signals. That is, melanin concentration are high in the case of control, whereas in the case of tyrosine knockdown, melanin concentration are very low, and in the case of this tyrosine re-expression, the melanin concentration further increases. And all these panels made a point that light scattering properties are related to the melanin concentration, and these light scattering properties can be used to identify the melanin concentration of the cells. So uh, this property they have used for their further studies in order to confirm uh, these melanin concentrations are present or absent within the cells, or are reduced or increased within the cells. So if you need to understand uh, in depth about this paper, you can go through the paper uh, or this study. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.